Summary of the genetic inheritance. The summary of the molecular basis of inheritance, right? So now the nucleic acids are long polymers of nucleotides, right? Nucleic acids, these are the long polymers of nucleotides. Now, while DNA stores genetic information, RNA mostly helps in transfer and expression of information, right? While DNA stores genetic information, while RNA mostly helps in transfer and expression of information. It transfer and expression of information, right? Though DNA and RNA both function as genetic material, but DNA being chemically and structurally more stable is a better genetic material. But DNA being chemically and structurally more stable is a better genetic material. However, RNA is the first to evolve and DNA was derived from RNA. RNA is first to evolve and DNA was derived from RNA, right? The hallmark of the double stranded helical structure of DNA is the hydrogen bonding between the bases from opposite strand. The hallmark of the double stranded helical structure of DNA is the hydrogen bonding between the bases from the opposite strands. The rule is that adenine pairs with thymine through 2 hydrogen bonds, guanine with cytosine through 3 hydrogen bonds. This makes one strand complementary to the other. The DNA replicates semi-conservatively. The process is guided by the complementary H bonding, right? A segment of DNA that codes for RNA may in a simplistic term can be referred as gene. So segment of DNA that codes for RNA may in a simplistic term can be referred as gene. So during transcription also, one of the strands of DNA acts as template to direct the synthesis of complementary RNA. So during transcription also, one of the strands of DNA acts a template to direct the synthesis of complementary RNA. In bacteria, the transcribed mRNA is functional, hence can directly be translated. In bacteria, the transcribed mRNA is functional, hence can directly be translated. Okay. In eukaryotes, the gene is split. The coding sequences exons are interrupted by non-coding sequences, that is introns, and the coding sequences exons are interrupted by non-coding sequences introns. Now introns are removed and exons are joined to produce functional RNA by splicing. The messenger RNA contains the base sequences that are read in a combination of 3 to code for an amino acid. So the messenger RNA contains the base sequences that are read in a combination of 3 to make triplet genetic code to code for an amino acid. Okay. The genetic code is read again on the principle of complementarity by tRNA that acts as an adapter molecule. So the genetic code is read again on the principle of complementarity by tRNA that acts as an adapter molecule. So there are specific tRNAs for every amino acid. The tRNA binds to specific amino acid at one end and pairs through hydrogen bond with codes on mRNA through its anticodons. So there are specific tRNAs for every amino acid. The tRNA binds to specific amino acid at one end and pairs through hydrogen bonding with codes on mRNA through its anticodons. The site of translation, that is protein synthesis, is ribosomes. The site of translation, that is protein synthesis, is ribosomes, which bind to mRNA and provide platform for joining of amino acids. One of the RNA acts as a catalyst for peptide bond formation, which is an example of RNA enzyme, that is ribosome. So one of the RNA acts as a catalyst for peptide bond formation, which is an example of RNA enzyme, that is ribosome. Translation is a process that has evolved around RNA, indicating that life began around RNA. So translation is a process that has evolved around RNA, indicating that life has begun around RNA. Since transcription and translation are genetically energetically very expensive processes, these have to be tightly regulated. Since the transcription and translation, that is transcription, that is making the copies of mRNA, translation, that is the synthesis of protein, that is protein synthesis, okay, are energetically very expensive processes. These have to be tightly regulated. Regulation of transcription is the primary step for regulation of gene expression. So regulation of transcription is the primary step for regulation of gene expression. In bacteria, more than one gene is arranged together and regulated in units called as operons. So in bacteria, more than one gene is arranged together and regulated in units, and they are regulated in units called as operon. So like operon is a prototype operon in bacteria which codes for genes responsible for metabolism of lactose. The operon is regulated by the amount of lactose in the medium where the bacteria are grown. Therefore, this regulation can be viewed as regulation of enzyme synthesis by its substrate. By regulation of enzyme synthesis by substrate. See, if more the substrate is present, lesser will be the formation of enzyme, and if less will be the substrate, more will be the formation of enzyme. So human genome project was a mega project that aimed to sequence every base in human genome. Okay, this project has yielded much new information. Many new areas and avenues have opened up as a consequence of the project. So DNA fingerprinting is a technique to find out variations in individuals of a population at DNA level. So DNA fingerprinting is a technique to find out variations in individuals of a population at DNA level. So DNA fingerprinting is a technique to find out variations in individuals of a population at DNA level. It works on the principle of polymorphism in DNA sequences. So DNA fingerprinting it works as a principle of polymorphism in DNA sequences. So it has immense application in the field of forensic science, genetic biodiversity, and evolutionary biology. Now, coming to the exercises. So group the following as nitrogenous bases and nucleotides. So adenine, it is a nitrogenous basis. Cytidine, it is it is nucleoside. And thymine, it is nitrogen base. Guanosine, nitrogen base. Uracil, nitrogen base. Cytosine, nitrogen base. Okay. So if a double stranded DNA has 20% of cytosine, calculate percent of adenine. So guanine is 30%, and cytosine is GC, right? GC. So guanine is 20%, and then 30% adenine, 30% thymine. Okay. If sequence of one strand of DNA is written as follows: 5 dash to 3 dash. Write down the sequence of complementary strand in the 5 dash to 3 dash. It has the same sequence as that of. If the sequence of the coding strand in transcription unit is written as this one, write down the sequence of mRNA. It is same, just replace the thymine, because thymine is not present in RNA. It is uracil. So AUGCA. So replace the thymine with by uracil. Okay. Now next thing is that which property of DNA double helix lead Watson and Crick hypothesis semi-conservative mode of DNA replication explain? Which property of DNA double helix lead Watson and Crick to hypothesis semi-conservative mode of DNA replication? To hypothesis semi-conservative mode of DNA replication explain? Because complementarity and a unique base pairing, right, and hydrogen bonding, and anti-parallel, and there is equal distance. So which property of DNA double helix lead Watson and Crick hypothesis semi-conservative mode of DNA replication? Because it is complementary in nature. Base pairing is complementary in nature. So explain. And they have the hydrogen bonding in between the nitrogen bases. So depending upon the chemical nature of the template DNA or RNA, the nature of nucleic acids synthesized from it, that is DNA or RNA, lists the types of nucleic acid polymerases. Depending upon the chemical nature of the template DNA, RNA, and the nature of nucleic acid synthesized from it, that is DNA or RNA, lists the types of nucleic acid polymerases. How did Hershey and Chase differentiate between DNA and protein in their experiment while proving that DNA is the genetic material? Because protein cannot replicate, DNA can replicate. Differentiate between the followings: and repetitive DNA and satellite DNA. So repetitive DNA, satellite DNA. Okay. mRNA and tRNA, that is messenger RNA and transfer RNA, also known as adapter molecule. Okay. Template strand and coding strand. So template strand, it is a DNA sequence, DNA strand, that is the DNA main frame strand, and then coding strand, but it does not code for anything. It is just named as coding strand. The naming is according with this reference taken from the coding strand, right? List two essential roles of ribosome during translation. So it can act as a enzyme, that is ribosome in bacteria, and ribosome is a synthesis of protein. So ribosome is a synthesis of protein, right? So ribosome is a synthesis of protein, right? Um, in the medium where the E. coli was growing, lactose was added, which induced the lack of iron. So in the medium where E. coli was growing, lactose was added. When the E. coli was growing, lactose was added, which induced the lack of iron, right? Then why does the lack of iron shut down sometime after addition of lactose in the medium? Because it's enough. They don't need the lactose anymore. So this is why. Explain in one or two lines the function of the following: promoter, tRNA, exon. So promoter where the RNA polymerase binds, right? Promoter. Then tRNA is transfer RNA. It takes the amino acid towards the mRNA. Then exon. These are coding sequences in the hnRNA, heteronuclear RNA in the eukaryotes. Why is the Human Genome Project called a mega project? Because it is 30 years of project and it involves lot of countries. This is a mega project and it's worth like $9 billion. What is DNA fingerprinting? DNA fingerprinting is based on the polymorphism and to know the variations at genetic between the individuals, the variations at the DNA level. Okay. So mention its application, right? It uses in forensic science, okay, and a paternity and all that. Briefly describe the following: transcription. It is the making the copy of an mRNA from template DNA strand. Okay, template DNA strand. So polymorphism, I told you, like variation at the DNA level. Translation, it is the synthesis of protein. Bioinformatics, a new era in the biology, that the study of the data, computers, and genetics. So this is it, your chapter.